Hello, today we will see a lecture on pairwise sequence alignment and also with a uh, online demo of Emboss tool. So I'm Dr. T. Anand. I'll be giving you this lecture. So what is pairwise alignment? Pairwise alignment is a sequence alignment technique. In bioinformatics, sequence alignment is a fundamental technique used to compare DNA, RNA, or protein sequences to identify regions of similarity. This may indicate functional, structural, or evolutionary relationship between the sequences. So the concept of pairwise alignment, we will be seeing about the concept of pairwise alignment, types of pairwise alignment, common algorithm used for pairwise alignment, and hands-on demonstration of using online emboss. In pairwise alignment, there are two major types. One is a global alignment, another is a local alignment. If you happen to do an end-to-end alignment between the sequences, that is sequences of similar length, then we would go about using Needleman-Wunsch algorithm or global alignment. But if you want to find regions of high similarity within a longer sequence and your query is a small sequence, then you will be using Smith-Waterman algorithm, usually a local alignment method. So both these methods follow what is called as dynamic programming approach for computing the alignment score. So let's see what is dynamic programming. Dynamic programming is an optimization technique used for solving problems by breaking them into smaller subproblems. It scores intermediate results to avoid redundant calculation, making it efficient for sequence alignment. Why dynamic programming in sequence alignment? Because this ensures an optimal alignment between sequences. And it also uses a scoring system to maximize matches and minimize gaps and mismatch. So this works efficiently even for long sequences compared to brute force method. So dynamic programming approach is the preferred choice of programming approach. So dynamic programming in pairwise alignment, there are two widely used dynamic programming algorithms for sequence alignment. One is Needleman-Wunsch algorithm, otherwise called as global alignment method. This alignment method aligns two sequences end to end, considering gaps whenever there is necessary. So this is for best used when we are comparing two sequences of similar length. And the other method is Smith-Waterman algorithm, which is otherwise called as local alignment method. This finds the high score on the local regions of the sequences. That is, if your query is of a short length and you are comparing against a huge database, this is the most preferred choice of algorithm. As I said, this is useful when sequences differ significantly in length. Okay, now let us see how dynamic programming works in sequence alignment. These are the major steps like initialization, then matrix filling, traceback. These are the three major steps. So under initialization, we usually create a matrix with the rows representing one sequence and columns representing the other. Once this step is done, we will go about calculating the score, assigning gap penalties along the first rows and columns. Once this step is done, we will fill the entire matrix using the scoring step. And the scoring function that is used is, example, a of I comma j is equal to maxima of a of I minus 1 comma j minus 1 plus match or mismatch. And the adjacent positions are a of I minus 1 comma j plus gap penalty and a of I comma j minus 1 plus gap penalty. So this formula ensures best possible alignment score at each position. Once this step is done, we will go about doing a step called as traceback, where we will migrate from bottom right corner, the top score, to the in a diagonal manner to the top left corner of the matrix, which is the minimal score. And this following this path of highest score will lead to the best possible alignment. Here is a visual representation of how the matrix is constructed. The right hand side you can see the completed matrix of how it will look. A left hand side is a calculation matrix where you can see there are two sequences, the top row and the column wise it is distributed. And so the first step, as I said, is initialization. The first box of the matrix is initialized with a 0. The first row and the first column is assigned gap, so they will have a negative score of -2, which gets incremented to the adjacent matrix. So you can see -2, -4, -6, -8, and -10, the adjacent cells. Same way in the column wise, you can see -2, -4, -6, and -8. So for explaining how this is done, let us look into one of the positions here. Here I'm going to explain you f of or a of 2 comma 3. So a of 2 comma 3, otherwise a of ai comma j is nothing but a of 2 comma 3 here. There are three scores that has been obtained from the adjacent cells. So first score is going to be -5. Let's see how it is obtained. It is nothing but maxima of a of I comma j minus 1 minus 2. So this particular position a of I comma j minus 1 is -3, and the gap penalty here is -2. So -3 minus 2 is -5. So which is what is this particular score. So if we happen to get this score, it is going to be -5. Similarly, from diagonally, if you happen to see, this is a of I minus 1 comma j minus 1 plus or minus plus match or mismatch. So which is going to be -1 here already, and because it is a mismatch here, which is again a -1, so -1 minus 1 is -2, so which is what is mentioned here. Similarly here you already have a score of +2. So this particular position a of I minus 1 comma j is +2. Because it is a gap that we are deriving, so it is a -2. So +2 minus 2 is a 0. So among these three scores, the maximum score is 0, which is what we are deriving here at this box. So if you happen to complete all the other matrix, you will end up having a matrix like this, where all the positions with the specific scores have been calculated and arrived at. So the bottom right corner, which you see the maximal score that has arrived. So in this case, 0 is the maximum score. So you will have to do a step called as trace back, where you will have to go to the next adjacent 3 cells, whichever is the maximum, you will have to go back. So here from 0, the adjacent cells, 3 cells are 0, 1 and 0. The maximum is 1, so we will go to the 1 cell. So trace back is such kind of method. So if you happen to look into next subsequent diagonals, you will see wherever there is a gap occurring, it will not go diagonal, but either go horizontally or vertically to the adjacent cell. So here in this position, you can see from 0 it is moving to 1. Again, we will have to go in the diagonal manner, and the same way here. Again, here at this position, you will see again it will move in the vertical manner, horizontal manner, and then again diagonally. So this is probably the best alignment that is possible for these two sequences. So here is how the output will look like. So here we have used a match or mismatch score of +1 and 0, and for gap opening we have used a gap penalization of -2 and gap extension a penalization of -1. So you will have an alignment like this where gaps will be inserted. So because gap is having higher penalty, although there are 18 matches, because of the penalization, you will end up having a score of +7 for these two sequences. Now in the next coming slide, I will be giving you a small demonstration of embossed Needleman-Wunsch algorithm to explain you how this is happening in an online tool. So let me play this video. So this is the embossed needle page where you will have to submit your sequence. This works on Needleman-Wunsch algorithm. I'm going to choose protein sequence here as my sequence of interest. So there are two boxes here you see where you will be submitting your query sequences, sequence 1 and sequence 2. And you can choose the output format in which you want the output. I want it in pad format. I'm going to choose alpha amylase as the sequence of interest. I've already searched this alpha amylase in NCBI, selecting protein sequence, and then I've searched for alpha amylase. I have arrived at two alpha amylase, one Homo sapien alpha amylase and Drosophila alpha amylase which I want to compare. I've already opened up the FASTA formats of these um, sequences. So that is your Drosophila sequence, and earlier you saw Homo sapien sequence. We'll copy the Homo sapien sequence first and then pasting it in the window. Here I've copied the Homo sapien sequence and pasting it here. Same ways I'll be copying the alpha amylase sequence of Drosophila and will be pasting here. Although they are both the same sequence, homologous in nature, you will see because of the dissimilar length, you will see the difference in result, and the percentage identity and similarity will be not very significant or very high. Let's view the result. So the result section will be something like this. It will have tool output, result files that you can download and visualize, and all the submission details that we have used for this particular program. Now the tool output, it will speak initially, it will speak about all the parameterization it has done, like how much is gap opening penalty, how much is gap extension penalty, and end open penalty, and all the other parameters that have been used by the algorithm. So at the bottom you can see the score, length that is compared 516, then the identity is around 274, similarity is around 348, and then around 27 gaps were you introduced into this alignment, because of which you can see the identity score is around 53.53% and similarity score is around 65%. We can refine it further to get maybe a better score. Now you can see there are in the graphical representation there are three symbols: a dash, a dot, and a colon. So dash is the region where there is a region of conserved nature. Okay, that is they are highly similar or conserved in nature. Then you have the colon, which is semi-conserved, and then the dot, which is partially conserved. So this is based on the physiochemical nature of the amino acids. It is, we can say it as semi-conserved or partially conserved. This scoring is got from the scoring matrices that is used here. In this case, I think we have used PAM 250 matrix, which is why it is giving you such a score. So this is how you do the emboss and get a pairwise sequence alignment. Hope this video was helpful. Thank you for your patient listening. Thank you. Let's see in the next video. Thank you. Thank you. Thank you for patient listening. Thank you.